In this video, we will discuss about how the organization in the endoplasmic reticulum happens, how different chaperones work, and their sequence. So this is the considered as a ribosome, and this is the protein that is translating inside the endoplasmic reticulum, and these are the translocon with the help of which this protein is translated inside. So as we know that this calnexin is a transmembrane domain and calreticulum is a soluble domain. So first thing that we need to understand that proteins or the chain of amino acid that is translating inside the endoplasmic reticulum has a more chance to interact with calnexin first and then with the calreticulum. What the studies have shown that if this NXT bond, this glycan structure is formed at NXT bond, NXS or T bond, and X should not be equal to P. X should not be equal to proline. N is asparagine, S is serine, and T is threonine. So if this this is triglycosylated glycan, as we have already discussed about this. So when this is then converted into monoglycosylated glycan. So if this monoglycosylated glycan is formed within the first 50 amino acid, then there is more chance that it will first bind to calnexin, and when the further glycosylation occurs in the later amino acid, then calreticulum will also bind to it. Calreticulum can also play a role in providing its native structure. And the third thing that we need to understand that if there is no carbohydrates, or this is also known as carbohydrates, if there is no carbohydrates binding occurred in the initial sequence of amino acid, then there is more probability than that this translating protein will bind to HSP chaperones, that is HSP 70 or HSP 90 or HSP 90, because HSP 70-90 will bind to the hydrophobic part of the proteins, and these glycans are hydrophilic. So there is more chance that HSP 70 or 90 will bind to the hydrophobic path. In that case, one more thing that people have understand that in order to confirm the studies that calnexin and calreticulum has some different properties, what people have done is that as calnexin is a membrane bound proteins, people have removed its membrane anchor and then study this calnexin function. Then they have found that calnexin behaves similar to calreticulum in the absence of this membrane anchor part of the calnexin. And similarly, if calreticulum is if in calreticulum someone adds the membrane anchor protein, then it will behave similar to calnexin. So in this way we can understand that calnexin and calreticulum are somewhat similar to each other, but they have some distinct properties. And calreticulum will is present deeper inside the endoplasmic reticulum because it is a soluble protein, whereas calnexin is a transmembrane protein. So there are more chances that calnexin will bind to the protein co-translationally than the calreticulum. And in eukaryotes, every protein on average has 500 amino acids. So it could take around 2 minutes for complete translation, for 2 minutes to complete translation. And within these 2 minutes, yeah, within the 2 minutes the monoglycosylation formation, monoglycosylation attached to this translating protein is very fast compared to this complete translation of the amino acid. So there is a good probability that calnexin, calreticulum, HSP70 play a role in co-translationally, and there are also evidence that some proteins have modified itself in a way that these chaperones helps them to attain their native structure. One of these example is of what's the example? One of these example, I don't have that document with me right now, but hemagglutinin. I might be wrong in the specification, but you will understand what is the essence of this thing. So hemagglutinin has cysteine, cysteine 14 and cysteine 466. This 14 is the number that is usually count from N-terminal side, because N-terminal is formed first and C-terminal is formed at the end. So this cysteine at 14 will bind to cysteine at 466 in its native form. So it has been assumed that with time this hemagglutinin proteins has been evolved in such a way that in order to avoid non-native formation of cysteine or the oxidation of cysteine, what happens is there are a lot of asparagine link, a lot of NXT sites. Asparagine link, NXT sites are available nearby this cysteine 14. So as soon as this N-terminal domain formed, it will get recognized by calnexin after monoglycosylation, and this N-terminal will remain close to this membrane. At the same time it will avoid the cysteine 14 to oxidize or make a non-native bond till the time this cysteine 466 is translated, and then it will help and after that this calnexin and calreticulum allows this cysteine 14 and cysteine 466 to make a native bond in an isolated environment, because after the complete translation of the amino acid chain, it will then move either to calnexin or calreticulum based on its substrate specificity, and then provide an isolated environment to make their native bonds. And there are also studies has been conducted for which it has been identified that calnexin has some specific substrate specificity and calreticulum has some different specific substrate specificity. For example, this proteins that membrane proteins such as vesicular VSVG. It is termed as vesicular stomatis virus G protein. It is a membrane protein and it is in the protein formation of this VSVG, calnexin play important role, and this has been confirmed with the help of by observing the cells with the calnexin knockout. By observing the calnexin knockout cell, by observing the VSVG protein in calnexin knockout cell, it will get affected, whereas in calreticulum knockout cell, VSVG proteins does not get much affected. And similarly for calreticulum, and for calreticulum, this hemagglutinin that we have discovered, this blood coagulation factor 5, coagulation factor 5, it is a heavy glycosylated soluble protein. This is heavy glycosylated soluble protein. So it is in the protein, in the native structure formation of this blood coagulation factor 5, calreticulin plays a major role. It is a heavily glycosylated protein, and when the number of monoglycosylated sites are more than 2, we have seen that this calreticulum will also bind to the proteins, and it is a soluble protein, and also it is a heavily glycosylated, so it is much more likely that calreticulum plays a major role in its protein formation, and this has been confirmed in case of calreticulum knockout. This blood coagulation factor is unable to form its native form, or its efficiency is decreased, whereas in calnexin knockout the efficiency does not decrease too much. So the sequence that organization that we can understand is, if there are no amino acids in the initial sequence, no amino acids initially, then this HSP 70 and 90 will bind. HSP 70 and 90 will bind. And if there are no amino acid initially, it means 50 amino acid, sorry, I mistaken, no glycan, no NXT site, no NXT sites in first 50, no NXT site in first 50 amino acid initially, then HSP 1750 will bind and helps the protein for its native structure. Whereas if there are 2 NXT sites in 50 amino acids, then it will binds to the, then calnexin will binds to this protein, and when the additional NXT sites are added, greater than 2 NXT sites, then calreticulum will also bind. Calreticulin will also bind and plays a role to get protein its native structure. So I think that is a good introduction about how the quality control or the process by which protein acquires its native form in endoplasmic reticulum works. Thank you.